Welcome to Biochemistry DSP channel. In this video, I'll be discussing about the composition of gastric juice and gastric juice analysis. Gastric juice is a colorless viscous liquid with a specific gravity of about 1.007. It is produced by the gastric mucosal cells and it is acidic in nature. Normally, about 2500 ml of gastric juice will be secreted every day by a person on normal diet, and a healthy individual can secrete about 1000 ml of gastric juice during the fasting state. The secretion of gastric juice will be due to the response of gastrin hormone present in the stomach, or it may be due to the psychological factors like sight, taste and smell of the food can also stimulate the gastric secretion, and presence of food in stomach, okay, that also can stimulate the gastric secretion. Amino acids like histamine will act as a potent stimuli for gastric secretion. Acetylcholine can also stimulate the production of gastric juice. So in the stomach, gastric secretion will be produced. It consists of about 99.4 percentage of water plus 0.6 percentage of solid. The solid constituents includes both organic as well as inorganic constituents. Okay, so organic constituents means mostly mucin will be present, pepsinogen, rennin. Okay, so rennin in the infant of stomach, infant gastric juice, no, it will be present rennin. In adult, it will be absent. Along with this mucin, pepsinogen, intrinsic factor, gastric lipase, gelatinase will be the constituent of organic constituent amount. Okay, then in the inorganic constituents means this gastric juice will contain H plus ions, chloride ions, sodium, potassium and small amount of phosphates. So all this will be the constituent of gastric juice. So gastric juice is acidic in nature. So we all know that acidic means the pH will be between 0 to 6. Neutral means 7, and above 8 to 14 means it is basic or alkaline in nature. Okay, so in the stomach you can the pH of stomach will be 2 to 5. Okay, so gastric juice which is produced in the stomach also will be acidic in nature. Okay, mechanism of HCl secretion. So how HCl is secreted in the stomach? Okay, so HCl is a main constituent in gastric juice. The secretion of H plus ions and secretion of chloride ions. This combination of H and chloride will be producing HCl. So in the presence of carbonic anhydrates, H2CO3 will be converted into CO2 and water in the parietal cell. This H2CO3 dissociates to form H plus ion and bicarbonate HCO3 minus. Okay, the H plus secreted in the lumen of parietal cell will be actually it along with the potassium ATPase pump. No, this H plus ions will be in it will be exchanged for K plus and chloride ions. Okay, so this bicarbonate produced or transported out through antiport mechanism with chloride. Okay, because of the high intracellular negativity, these chloride ions are forced out through chloride channels on apical membrane. Okay, because of this high intracellular negativity, sodium potassium pump, which is located at the basolateral membrane, will pumps out 3 sodium ions for every 2 potassium ions. Okay, so this K plus pumped will diffuse out through K plus channels present on the basolateral as well as through apical membranes. Okay, then now we have to know two views are seen in HCl secretion, that is Davenport view and modern view. So according to Davenport's view, uh, a high energy compound is oxidized. Okay, during electron transport chain, what happens means a high energy compound will be get oxidized to yield H plus ion. So for HCl secretion, how we get H plus ion? It can be connected with two views. Okay, in terms of two views, we can connect the formation of H plus ion. The electron is processed in the mitochondria by the cytochrome system and is accepted by oxygen. Okay, this is in the during electron transport chain. This process will be there. During this process, OH is formed and energy will be liberated. The H plus ion formed initially uses the energy liberated to form HCl. Chloride for this reaction comes from plasma. Okay, this is according to Davenport's view. According to modern view, H plus ions in the parietal cells are obtained by active ionization of water, which yields H plus and OH ions. OH should not be allowed to accumulate inside the cells. Hence, it is neutralized by the manner that is CO2 produced inside the cell will contribute from the plasma. With the help of carbonic anhydrates, it will be converted into H2CO3. This H2CO3 will dissociate into H plus ion and bicarbonate ion. This H plus ion will neutralize this OH ion. Okay, so H plus and OH will react to form water. So in this way only we will get this H plus ion. Okay, then this is the mechanism. Okay, mechanism of HCl secretion. So through bicarbonate, through carbonic anhydrates, this H plus and HCO3 minus ion will be split up. Okay, then chloride also will enter. This chloride will react with the H plus ion. Then it will produce HCl. Okay, finally we'll get HCl. This is how HCl will be secreted. So actually this carbonic anhydrates is the main thing which is responsible for this HCl secretion. Okay, so if carbonic anhydrates is blocked or inhibited, the entire HCl secretion also will be blocked. Okay, the H plus ion formed because of ionization of water is pumped into intracellular canaliculus in exchange for potassium. This H plus K plus pump activity requires the energy supply. Okay, now the HCl secretion is inhibited by so many factors like acid in the pyloric antrium, then acid in the duodenum, fat in the duodenum, hyperosmotic particles in the duodenum, emotional factors like depression, disappointment. All this will inhibit the HCl secretion. Okay, so there are some stimulant and depressant. Stimulant may be parasympathetic nerves, acetylcholine, gastrin, histamine, calcium ions, caffeine, alcohol, nicotine, protein, food flavors, medications. Okay, so all these will act as a stimulant for HCl secretion. Then presence of depressants like fat, acid, alkali. Okay, parasympathetic drugs, proton pump inhibitor, prostaglandin, stomatostatin, all these will act as depressants for HCl secretion. The role of HCl is to activate pepsinogen into pepsin. Okay, it has to convert the zymogen, inactive enzyme pepsinogen, into an active enzyme pepsin. It is necessary, this HCl is necessary to maintain optimum pH for pepsin activity. It also acts as a bactericidal effect, ion absorption. It stimulates bile and pancreatic juice secretion. Now we'll come to gastric juice analysis. Okay, we have seen what are the constituent organic and inorganic constituents of gastric juice. Now we have to know why this gastric juice analysis has to be performed. Okay, so this gastric juice analysis is performed to diagnose certain diseases or disorders in the stomach and duodenum. That is the main reason why this gastric juice content has to be analyzed or examined. Okay, we have to analyze both physical and chemical examination of the gastric juice to get some informations about the diseases in the upper gastrointestinal tract. Okay, so mainly the diseases related with the gastrointestinal tract includes gastritis, then gastric ulcer, gastric carcinoma, TB of stomach, hyperchlorhydria, pernicious anemia, Zollinger Ellison syndrome, duodenal ulcer. So these are the uh, diseases which can be diagnosed with the help of gastric function test or gastric juice analysis. Okay, These are the main tests will be included under gastric function test. That is examination of resting gastric content, that is gastric residuum. Then fractional gastric juice analysis, fractional gastric juice analysis after administration of a stimulant. Okay, mainly they will perform all these tests. Now in this video we'll see only about the first test alone in this video, and in the following videos we'll discuss about other gastric function test. Okay, so now we'll first, before going into the test, we have to know how to collect gastric juice. So in commercially there are there are so many tubes and tubes available to collect the gastric content. That is Ryle's tube. Okay, so these are tubes which are introduced into the stomach. Okay, Ryle's tube, Rehfuss tube, Levin's tube. Okay, all these tubes will aspirate the content of stomach with the help of a syringe. Okay, so through nasogastric route we have to insert this tube into the stomach and aspirate the gastric content with the help of syringe. Okay, the stomach content collected after an overnight fast is called gastric residuum. Okay, so this is the name, examination of resting gastric content. That means what? We are collecting the gastric juice after an overnight fast. That content, uh, collection is called as gastric residuum. Okay, it involves then this gastric content what we have collected, no, that has to be examined for both physical and chemical examination. Okay, chemical, that is physical examination means we have to check for the color, odor, volume, and any deposits are present, or for the consistency of the gastric juice. Chemical means we have to examine the acidity, bile, digestive enzymes, presence of digestive enzymes like pepsin and rennin, blood and organic acids. So we have to chemical examination means we have to analyze certain qualitative test. Okay, and physical examination means we have to physical appearance of the gastric juice. So we have to check for the color, volume, smell, consistency, and deposits. Okay, chemical examination means we have to do the test for the presence of organic materials and inorganic materials. Okay, so mainly organic materials means it will have protein, pepsin, then mucin. Okay, it's a carbohydrate. Glycoproteins will be present, HCl will be present, lactic acid. Then abnormal constituents like blood also will be present in the case of any disorders. And for bile and bile pigment also we have to check it. Okay, color. So physical examination first, we have to check for the color of the gastric juice. Normally the gastric juice will be clear and colorless. Okay, it may be slight yellow or green due to the presence of bile regurgitated from the duodenum. Okay, a dark red or bright red or reddish brown or coffee ground gastric juice indicates the presence of blood. Okay, blood, mainly it occurs in the gastric juice due to peptic ulcer, gastric carcinoma, then any trauma of the gastric mucosal cells. So on seeing the color of the blood itself we can see, okay, we can go for the diagnosis. Okay, so the color of blood will be coffee ground or reddish brown, means it may be due to the formation of acid hematin in the stomach. Mainly this coffee ground or reddish brown gastric juice will be observed in the case of gastric carcinoma and peptic ulcer. Bright red color means it indicates the presence of fresh blood. Okay, some damages in the mucosal cells or ulcer conditions, we can see a fresh blood leaked into the gastric juice. Volume. Okay, normally the volume will be about, we can collect about 20 to 50 ml of gastric residuum. An increase in volume of greater than 100 ml indicates hypersecretion of gastric juice or retention of gastric contents over due to delayed emptying of stomach or due to some regurgitation in the duodenal secretion. Okay, a decrease in volume less than 20 ml will be seen in the case of pernicious anemia. So these are the changes we can come across with volume. Okay, then odor and smell. Normally the gastric juice will be odorless. A rancid or foul smell is noticed, means it indicates gastric carcinoma, gastritis, malignant stenosis. Ammonia smell may be due to uremia. Then consistency and deposit. Okay, so normal gastric juice will be a fluid in consistency. It will not be viscous. It will be like a fluid, clear fluid with no deposit. If some food particles are present, means it may be due to gastric carcinoma, pyloric stenosis, pylospasm, etc., or pulse cells, tumor cells, epithelial cells, and RBCs will be also observed in the case of gastric carcinoma. That's all about the physical examination. Then chemical examination means we have to check for the normal and abnormal constituents of gastric juice. We have to do some qualitative test to find out the presence of normal and abnormal constituents. So if you are not detecting any abnormal constituent, means the person is normal. If the person's gastric juice is found to contain abnormal constituents like blood or any abnormal unwanted materials, means it indicates some sort of gastrointestinal problems. Okay, so the normally carbohydrates, that is glycoprotein will be present, that is mucin is present as a main constituent in the gastric juice. So mucin means it is a glycoprotein, so it will give positive result for biuret test because it's a protein. Okay, mucin is a glycoprotein, so protein will answer for this biuret test. And carbohydrate, glyco means carbohydrate, so Molly's test will indicate the presence of carbohydrate. Okay, so it will indicate the normal amount of mucus cells. Okay, normally it will have small quantity of mucin, and increased level of mucus also may be due to gastritis and carcinoma of stomach. Then for the presence of HCl, you have to check for the free acidity by using indicator, Rehfuss indicator test. Okay, you have to take 2 ml of gastric juice in a test tube and you add 1 drop of Rehfuss indicator. This Rehfuss indicator is nothing but dimethyl amino asobenzene. Okay, so presence of HCl will be indicated by the appearance of red color. So in the near the pH 3, no, this Rehfuss indicator near we will give pH 3. That time we will get a red color. It indicates the presence of HCl. Then Gunsberg test. In this method, you are going to evaporate 1 drop of gastric juice with 1 drop of Gunsberg reagent. A formation of red color crystal or paste will indicate the presence of HCl. Okay. So the following things can be you can check for the with a test of HCl. Okay. So normally gastric juice contains HCl. If there is no secretion of HCl, means the condition will be called as achlorhydria. Uh, okay. No, completely HCl secretion is absent, means achlorhydria. Then less secretion of HCl means hypochlorhydria, and more secretion means hyperchlorhydria. Okay. In achlorhydria, we can find there is complete absence of HCl, but enzyme pepsin will be present. Okay. Whereas if both HCl as well as this pepsin are absent in gastric juice, means that condition is called as achalia gastrica or gastric achalia. Okay. Then this condition, this achalia gastrica condition will be observed in the case of TB of stomach, ulcerative colitis, then pernicious anemia. Mainly in older age people, we can above 70 years, no, we can observe this achlorhydria problem. Then hypochlorhydria means less secretion of HCl. That is, the HCl level will be below the normal range. It will be observed in the case of carcinoma of stomach, gastritis, constipation. Hyperchlorhydria means increased secretion of HCl, free acidity of gastric juice. That is, it will contain 100 milliequivalents of HCl. So that is the free acidity level value will be more than 100 milliequivalent per liter in the case of hyperchlorhydria. It will be seen in the conditions like gastritis, cholecystitis, and duodenal ulcer. Blood along with this hyperchlorhydria will indicate gastric ulcer and also in gastric carcinoma. Then the other chemical test will include about lactic acid. Okay, for lactic acid we can perform ferric chloride test. For this ferric chloride test, you have to take 10 ml of distilled water in a test tube. You have to add 10% ferric chloride solution. observe for the canary yellow color. That will indicate the presence of lactic acid, which is organic acid. So organic constituent lactic acid is present. Okay. So lactic acid is a important organic acid which is present in achlorhydria. Okay, or during the retention of food residues, no, we can find more amount of lactic acid present in the gastric juice. Okay, mainly achlorhydria will be associated with retention of food particles in the gastric carcinoma. Then test for acute blood. So for this benzidine test will be helpful. You have to take a pinch of benzidine powder, add 2 to 3 drops of glacial acetic acid, mix well. Then add hydrogen peroxide, 2 to 3 ml of hydrogen peroxide. Then add 0.5 ml of gastric juice. Okay, a faint green to blue color will develop in the top of the test tube. That will indicate the presence of blood. Presence of blood in gastric juice will indicate peptic ulcer, gastric carcinoma, trauma of gastric mucosal cells. Okay. Then test for pepsin. For this, you have to take 5 ml of milk with 5 ml of buffer in a test tube. Add 1 ml of gastric juice. Keep it in water bath for 37 degrees Celsius. You have to check for the curdling of milk. So normally, curdling of milk occurs means it indicates the presence of normal constituent pepsin in the gastric juice. If absence of pepsin, no curdling occurs means it indicates the absence of pepsin in the gastric juice, which is seen in Echinacea gastrica. Then test for bile and bile pigments. Of presence of bile can be detected by performing Hayes sulfur powder test. Okay, this is a specific test to detect the presence of bile salt. You have to take 5 ml of gastric juice, sprinkle some amount of dry sulfur powder on the top of the gastric juice. You have to check for the position of sulfur powder. So if the sulfur powder sinks to the bottom, uh, it indicates the presence of bile salt. Okay. Then presence of bile salt in gastric juice, also it can also be detected with the help of bile pigments like Gimmelin's test or Foucher's test. You can also perform Gimmelin's and Foucher's test to detect the presence of bile pigments. Okay, these are the chemical examination, that is qualitative test we can do to examine the gastric juice. Then quantitative estimation. So what quantitative estimation you can check with this gastric juice means you can check for the free and combined acidity. So for this you can do a titration. Okay. So the basic principle involved in this quantitative estimation is the main acid present in gastric juice is HCl, that is a free acid. Okay, you are going to in addition to this acid, we have got some of the organic acid like lactic acid and pyruvic and butyric acid is also produced in the stomach. Okay. So, this normality of this free acidity and combined acidity can be determined by titration, by titrating these acids against sodium hydroxide using two type of indicators like Toffus indicator and phenolphthalein. Okay. So this is the principle involved in this quantitative estimation. If you add up this both values of free acidity and combined acidity, it will give the total acidity of gastric juice. So two types of estimation you will be performing. First, in the estimation, you will be determining the free acidity. That is, you will be calculating the strength of HCl. So it will be indicated this HCl will be titrated against 0.1 N sodium hydroxide using Toffus indicator. So the Toffus indicator will act in the pH range of 2.9 to 4. Okay. The color changes from red to yellow. Okay. That will indicate the endpoint of the titration. So here the first you have to estimate the free acidity. So in the for doing that free acidity, you have to take a 0.1 ml of sodium hydroxide in the burette. Take 10 ml of gastric juice in the conical flask. Add 1 drop of Toffus indicator. After adding Toffus indicator, bright color appear, red color appears. Okay. You have to titrate this conical flask against this, that is gastric juice, against this sodium hydroxide solution till that bright red color turns into orange yellow color. Okay, that is called salmon yellow. That is the end point. Okay, you have to note down the reading of this end point. Then you have to go in for the determination of combined acidity, the next titration. So now this bright red has been turned into orange yellow, the first after that. Okay, now in that solution you add 1 drop of phenolphthalein indicator in the conical flask, titrate against sodium hydroxide till that okay, that uh, a pink color, okay, faint pink color appears. That will be the end point. Okay, till the color changes into a pink color, that will be the second end point. Again, note down the value, calculate, do the calculation and check for the values for free acidity and combined acidity. Okay, so normally the free acidity value will be around uh, actually it will be around 15 to 35 milliequivalent per liter, and uh, total acidity will be about 25 to 45 milliequivalent per liter. Okay, so suppose if you are getting in this titration well method, if you are getting free acidity as 30 milliequivalent and combined acidity as 12 milliequivalent, means the total acidity will be the sum of free acidity plus combined acidity. It will be about 42 milliequivalent per liter. Okay, so this is how you have to do the quantitative estimation of free and combined acidity of gastric juice. Okay, so these are the tests. Okay, this is the first part of test to be done with resting gastric juice. Okay, so we have to do three types of gastric juice analysis. This is the first method. Okay, we are going to examine the resting gastric content and gastric residuum. Okay, in the fasting sample we are going to take it. Okay, so in this you will be doing physical examination, chemical examination and quantitative examination of free and combined acidity of gastric juice. In my next video, I'll be explaining about the other gastric function test. Thank you for watching this video. Subscribe and support the channel Biochemistry DSP.